In this video, I will explain what an inhibitory neurotransmitter is. Here we have a nerve to communicate with the effector cell, which in this scenario, the effector cell would be neuron or muscle cell. The nerve releases neurotransmitter molecules. Then these molecules should diffuse toward the membrane of the cell that we have here. In the membrane of the cells, we have receptors. What binds to the receptors would be hormones and neurotransmitters. Each cell has a variety of receptors for different neurotransmitters and hormones. However, in this image, I show you the receptors only for this particular neurotransmitter. So the goal is that the neurotransmitter gets here and binds to the receptor. But first, we take a look at the receptor in a closer view. These receptors that we are talking about are part of an ion channel. Currently, the gate is closed because the opening of this ion channel happens exactly when the neurotransmitter binds to its receptor. And recall that we named the ion channels that opening their gate depends on binding a molecule to them, ligand-gated ion channels. Then we take a look at the membrane of the cell and measure the voltage. Since the ligand-gated channels are closed currently and no ion diffuses in or out, let's say the voltage of this neuron or muscle cell is at rest. The question is, when the neurotransmitter binds to the receptor and opens the ion channel due to the diffusion of ions, what happens to the voltage? Does the voltage goes up toward the threshold, that would be depolarization of the cell, or the voltage starts moving away from threshold, that would be hyperpolarization of the cell. So let's continue the story and see which one of these two is the scenario here. One scenario is if the receptors are part of potassium ion channel. When the neurotransmitter binds, potassium ion channel opens, and since we have a higher concentration of potassium ion inside the cell, this ion starts flowing out, and that leads to hyperpolarization of the cell. When we follow the voltage in the membrane of the cell, we notice that the voltage starts moving farther away from threshold. That simply means the cell is inhibited. The other scenario is that the receptor would be part of chloride ion channel. When chloride ion channel opens, since we have a higher concentration of chloride ion in the extracellular fluid, or we can say interstitial fluid, chloride ion, which is a negative ion, starts flowing into the cell, diffusing in. And this also leads to hyperpolarization of the cell. So the conclusion that we make is this. If a neurotransmitter molecule open potassium ion channel or chloride ion channel, opening either one of these channels leads to hyperpolarization of the cell. Cell is inhibited. That's the main reason that we named the neurotransmitter molecules that open either potassium ion channel or chloride ion channel inhibitory neurotransmitters.